is really the exciting news for the whole month of May. People probably already saw from New York Times, Endpoints News, Stat News, everywhere. This is really probably because the whole field of CRISPR as a therapeutics has been struggling for the last year. As people know, people even using the terminology, there's a CRISPR bubble in the biotech industry. And especially with those CRISPR delivered by AAV, now the AAV gene therapy has its own struggle. So the whole cell and gene therapy, I would say on the gene therapy side, there's a lot of kind of second guessing or ranking about what's going on in the field, especially in this day and time when biotech venture money has become very tight. But I think this latest news from Children's Hospital of Philadelphia really encouraging. That represent probably the major piece of good news to the whole CRISPR field, although it's using the base editing, but the base editing is still based on CRISPR original technology. So I really want to cover this topic in this month's Biotech News. And I probably think 15 minutes won't do its justice. Probably, yeah, that's why I can only kind of condense my presentation. In the future, we can have more discussion as this patient, whose name is KJ, is keep doing well. Right now, he's at 9.5 months old, close to 10 months. If he keep doing well at 1 year, 2 years, I think there will be a chance for BioSpark to even dive deeper in terms of personalized gene editing therapy for those rare genetic disease. So here is just a quick outline in terms of time. It just tells you how amazing this story. When I was reading the New England Journal of Medicine paper, as well as the report, it's more exciting than novel. Because when you're reading a novel, a fiction, usually it's a made-up story. But this is real story, and with a baby's life on the line. So that's why making the story even a little bit emotional, I have to say. Even I'm not a person that easy to be emotional. But in terms of the diagnosis of the disease, this kind of disease was Children's Hospital of Philadelphia, CHOP, is a very well-known children's hospital across the whole world. And this particular collaboration between two PIs, first also is Dr. Musunuru, and the last author, they have been collaborating. They were thinking about how can we use this CRISPR or base editing related technology to helping the patients. And they really been testing that because they usually, when you have a CRISPR design, usually there's not enough time to have a personalized therapy ready. So they've been testing for quite a number of years, 2 or 3 years. It's not like this is the first patient ever being tested. So they named their patient from a Greek letter, alpha, beta, gamma. So by this patient we're talking about today, it's already their patient number 7. So the first 6 patients, they have to pass. They decided our turnaround time in the CHOP was not ready. We cannot turn around to design a guide RNA, to have the LNP-formulated drug product ready for clinical testing. So usually they don't want to give patients some false hope. So they were able to finally gain some confidence, but it's still gamble because you are running against the clock. So the patient was born in August 2024, named KJ. And by the time of 48 hours after birth, there's something already going on with this particular infant because there's definitely a lot of abnormality. But diagnosis won't be as easy. There's a lot of possibilities. There could be sepsis, could be some metabolic disorder. Because CHOP has the capability to do whole genome sequencing, so they immediately sending the patient's blood sample and extract the genomic DNA, sending for sequencing. It takes a while to get the whole genome sequencing done. But during the 48 hours, I think one of the nurses really, based on her rich experience with many disease, pediatric disease, she has the insight saying, something's going wrong. Why don't we testing this patient's blood ammonia concentration? So that turned out to be a very important idea. So once they tested the ammonia concentration, it's just off the chart. Then you know something is going on with a particular disorder called urea cycle. I won't have time to dive deep into the deep history of metabolic biochemistry. Actually, the urea cycle is a very important disorder. And let's move to the next slide. And it's discovered by a probably one of the most greatest biochemists in the 20th century, Hans Krebs. People know the Krebs cycle, TCA cycle, or citric acid cycle. That's only his second cycle discovered. He and his graduate student first discovered urea cycle in 1932. So that's why making him one of the greatest biochemists in metabolism. And the urea cycle in a webinar, people interested, you can, on the lower right corner, I gave a webinar on Nobel Prize prediction in 2022. At that time, it's very coincidental. That's why always fascinating to see many different fundamental biology or cell biology can come together into some common thread. And in that webinar, I was predicting the chaperone, molecular chaperone work will eventually get a Nobel chemistry prize. It has not happened yet, but I think I still hope sooner or later it will happen. So for those native Chinese speakers, you can scan the barcode if you were interested in the history. So one of the key leading figure in the molecular chaperone field, Arthur Horwich, got his independent career started at Yale as a pediatric resident. And he happened to seeing some patient with urea cycle defect. The urea cycle showing on the left and involving 5 major enzymes, not all discovered by Hans Krebs. Hans Krebs and his graduate student only described the big picture in terms of it's a cycle, but individual reaction require additional scientists to contribute. So the first enzyme in this cycle is called CPS1. That's the rate limiting step. So what is urea cycle really doing is to convert toxic ammonium. NH3 ammonium is very toxic in the human, especially to the new nervous system. So you have to make sure the amino acid degradation leading to the ammonium or other sources of ammonia has to be converted to a less toxic urea. That's why urine is named essentially after urea. So urea will take in 2 nitrogen and combine with the carbon dioxide and into the urea. Then this is less toxic. It can be secreted through urine. So this is a very important metabolic step for the whole body to make sure the amino acid breaking down won't become a toxic burden, and as well as helping many other things. So the urea cycle, because it's important to the liver function, and the liver is really carrying out the metabolism urea cycle, it becomes very important, especially for a newborn baby, to control the ammonium concentration. So in this webinar 2022, it's just Arthur Horwich happened to interest in the second enzyme, OTC, ornithine transcarboxylase. That's also a mitochondrial enzyme, but in this case, the Children's Hospital of Philadelphia, this baby having 2 mutations in the CPS1 gene. And both are nonsense mutation, a stop codon, one from father, one from mother at different location. One is at the Q335, one at E7114. And both a single nucleotide point mutation leading to a nonsense stop codon, which leading this baby have no functional copy because the father's copy and the mother's copy each are truncated protein, or even going through nonsense-mediated decay for the mRNA. So it's very kind of interesting situation. It's a compound heterozygosity, and it's a life-threatening because if you're not careful controlling the ammonium concentration, the neural damage, brain damage will be permanent. So they have to using scavenger in the ICU to make sure the ammonium concentration won't be over the chart. Then go to next slide. I will talk about why they chose this base editing. Because base editing was invented by David Liu around 2015 or even potential 16, around that time after the CRISPR has been invented. He was able to re-engineer the Cas9 protein, making it a nucleus dead. So it's called a dCas9. But he also has the insight because his chemistry background, he's a very deep chemistry background. He will try to fuse in a deaminase domain with the nucleus disabled Cas9. And then this can, using the guide RNA, to bring the whole complex onto the target DNA. And finding near the PAM region, PAM stands for protospacer adjacent motif, where how the CRISPR is working, rather than cutting the DNA into double-strand break, the deaminase can find the right nucleotide or the base. There's only 2 types of base editor. One is called the Adenine Base Editor, ABE. One is called cytosine Base Editor. So that's why it's representing about 30% of the point mutation that can be addressed by base editing. So that's why David Liu in this year got the breakthrough prize for the invention of base editing. I will go into more detail on the next slide. So it turns out that because you're limited about the ABE and the CBE, in this baby's case, its coding strand C2T mutation happens at the glutamine 335. So that means you can, using the ABE, because on the antisense strand, you can convert the A back to G, but it's a multi-step. A has to be deaminase to be inosine. Then inosine will using the DNA repair mechanism to become a G. So reverting a back to G, so that the genetic codon will no longer be the stop codon. It will reverting back to the glutamine Q. So, but the other one from the mother, E714X, cannot be corrected by this kind of base editing. So that's why eventually that one was not used. They're only fixing the father's copy, which happens earlier in the protein level. So I won't go into the details about base editing. People can read the literature, but essentially it's using the CRISPR mechanism and finding the target nucleotide for base editing. But sometimes the neighboring base will also be base edited. So that one, you require some luck. Let's move to the next slide. And we're talking about how do they come up with this very bold strategy to do a personalized, because this is a unique mutation for this particular patient. So in order to even screening for the best combo of base editor with the guide RNA, ideally you want to do it in primary hepatocyte, but the baby is too small. It's very unethical to try to get some hepatocyte from this baby. So how do you do it? That's why they have, for many years experience, they already having this system designed. They're using a hepatomer cell line, similar to liver, but the hepatomer cell line don't have this baby's mutation. You have to knock in. But do you knock in the whole gene? Because this is a very huge gene, 1,500 amino acid. So they very innovatively only design a 100 base pair nucleotide centered around that point mutation, but they also including as a control for the E714X. Then they also adding some phenylketone urea, which they know in the lab, they can cure with base editing. So as to another positive control. So it's a cassette of 4 100 base pair nucleotide, because base editing happens on single strand RNA, a single strand DNA. And that's why they having this staggering as an assay system. You have to using lentivirus to transduce this cassette into the hepatocellular line so that you have the target for base editing. So that's why it's a very innovative approach, but they also need to design staggering guide RNA showing on the top right. They have about 9 guide RNA staggering around this Q335X mutation. And this graduate student, Sarah, the second author on the paper, really working day and night for 1 month to getting the kind of cell line established, transducing. So then spend another month, were able to finish the screening and finding a combo of one of the ABE editor shown on the lower right corner. And basically, because they're using a different engineered Cas9, allowing flexibility of not NGG PAM, they're allowing to do NGC PAM. Then you have the off-target problem because it increased the off-target effect. Then the protein engineer found that V106W, if you introduce that, it can somehow offset the off-target problem. But they have to do the screening. So luckily, by finding their ideal ABEA1 showing with the red arrow, combined with the mutation in Cas9 V106W, it only decreased the editing efficiency a little bit. It's still above the red line. Whereas the other combo, once you introduce the V106W, the editing, base editing efficiency dropped dramatically. So to their luck, they were able to find a combination of guide RNA number 8 with the base editor they want, having a mutation lower the off-target rate. So that's why it's a very impressive luck you have to have. Even more than that, if you look at the lower right corner, and the target base is the red color, 335X. That's the target A8 you want to convert back to G. But it turns out some neighboring G were also affected. Some neighboring A, there's other A's like this A12, A9, A3 were also affected. So that's the off-target. That's called a kind of bystander editing. And also to their huge luck, if you look at the genetic code, all those bystander A to G editing happen on the third position of genetic codon. People remember Francis Crick, the wobble hypothesis. If you only change a nucleotide on the third position, usually the amino acid will be synonymous. So to their, all the star align for them, all the bystander editing does not change the amino acid. So if you ask me why the other A does not get edited, nobody can answer, because we still don't know the molecular detail base editing. It just happens through empirical sequencing. Only those A bystander editing, but all of them are synonymous. You need some luck in science. So let's move to the next slide. So they found this ideal combo, then they just using some very hard work in terms of you have to transduce the same cassette using the knocking transgenic mice, knocking to the mouse model, and then working with Jackson Lab to eventually coming up with a in vivo dose response efficacy or potency. It's not really clinical efficacy, it's for potency testing. And to see that this particular combo, what they called KABE, is named for the drug product, were able to show the in vivo efficacy in the mouse model. And then they also need to addressing FDA's potential question for off-target effect. That's why in this case, they were using some human primary hepatocyte to see whether the wild type CPS1 will, not only the CPS1 may be affected, could also affect other genes. Then it's relatively clean if you show on the lower left corner. And they use one is their HUH7 cell line they use for the in vitro screening. Then they all have 3 different donors for the hepatocyte, and that's different color. You see mainly you see the on-target effect, which is the lentiviral and the CPS1 showing the edit. But all the other nominated site by 3 different technology, 1-seq, change-seq, base editing, and guide-seq, all those additional 21 sites, they don't see evidence of off-target base editing. So that's why it's a pretty clean in terms of combination. Let's move to the next slide. So then they starting to work very hard, because this one, I think when you're reading the paper and the news report, it takes 3 companies to help them probably without asking for any pay. So one company is called IDT in Iowa, whose strength is oligonucleotide related to CRISPR. They put 12 co-authors on the New England Journal paper. Those 12 FTE work around the clock to manufacture all kinds of guide RNA from guide RNA 1 to 9. Then eventually, once you settle on guide RNA 8, the Iowa scientists have to work around the clock to manufacturing large amount of quantity available. Then they ship to the guide RNA, clinical grade guide RNA to North Dakota. Well, Aldabron, the Canadian CDMO, Aldabron is based, they're manufacturing US. They will receive the guide RNA. They also will generate the mRNA. Why you need mRNA? All the nuclease, the nickase, and as well as the nuclear localization signal, the CRISPR enzyme, really, they put into an mRNA drug product. So the guide RNA and the mRNA product has to be assembled at Aldabron site, and but they need LNP. Lipid nanoparticle is not what Aldabron and IDT are familiar. So Acuitas from Vancouver has to send 2 FTE without pay to Aldabron's North Dakota site, working around the clock to formulate the LNP, putting this together, and so that they can have some clinical grade drug product in LNP formulation ready for KJ to be dosed. So this is just a kind of history-making type of efforts across academia, and David Luce people putting all the postdoc and all these efforts putting together can make this happen. It's a kind of amazing story. I highly recommend people to reading the whole story. I won't go into too much detail, but just suffice to say, the baby was born in August. Within 2 months, they get a combo. By 3 or 4 months, they finish the mouse efficacy testing. Then they're using non-human primate, wild-type monkey to test the LNP safety. Then they choosing the dose not based on mouse model, choosing based on the LNP safety batch, and choosing the dose very carefully. So by February 2025, about 6 or 7 months of the baby, he can be transfused intravenously with this drug product. And it's just amazing story. And you can see they didn't do a full panel testing, just testing the baby. And this baby, if the gray bar here on the left, you see already having some disease, because those babies are very vulnerable to any viral infection. Before he was receiving dosing one, at 140 days of age, he having some infection. Then the physician was able to manage that. Then after the dosing in February, he having 3 or 4 different viral infection. But because his condition improved so much, because he can reducing the, I don't know, people can read, you need a drug to manage the ammonium concentration. It's called the glycerol phenylbutyrate. So the dose before the treatment is about 10 ml per square meter. It reduced the dose by half. That's already tell you the base editing seems to be working on the CPS1. Then you look at the plasma ammonium concentration in panel B. Before the dosing is very much fluctuation. Every time it fluctuates up, you have to rush into the ICU to really do a lot of management. But after dosing, you can see the blood plasma ammonium concentration has pretty static. It's just very amazing. Then they're starting to give the baby regular protein diet after the second dose, so that you can giving the baby normal nutrition, and while still maintain the ammonium concentration. The urea cycle seems to be working, but they're still very conservative. They give the second dose about 4 weeks, 3 to 4 weeks after first dose. Then recently, after news is published, the baby received the third dose, because they have to be sure that the LNP does not cause toxicity. Then you can see the plasma ammonium concentration before and after has been trending down. And another biomarker, uric acid, is tell you if CPS1 is really working, it will produce some uric acid in the uric acid in the urine, then it will also trending up. That means CPS1 is really coming back. So it's just quite amazing story. So I think I can wrap up. Next slide is just the one to finally wrapping up, saying the story does not end here. We are at the month 8 or 9. The baby already got the third dose. Hopefully, he no longer need another dose, but it will be continued monitoring. That's why it's just an amazing story. So here I just stop. I'm already over time, and I would recommend people can type their question in the chat.
So Ethan, if this therapy get approved someday, does the company have to get IND approval from FDA for every patient?
Yeah, that's a very good question. Actually, Peter Marks wrote an editorial along this article. Actually, Peter Marks, during the time he was leaving FDA, I think that's something he feels very proud. He made a very good point. So probably it's not a good idea for every time you have to apply for IND for individual patient. He proposed that a company can take this over and file some IND for the backbone, for the mRNA backbone and the LNP formulation, so that whatever new disease, you just insert a cassette into the backbone, so that you apply patent protection and FDA approval for the mRNA backbone. Then it can fit into different disease using the same IND application and FDA designation, so that by working on different disease individually, you may be able to make some money. That's why I think for this to be commercially viable, FDA has to work out some new regulation.
But FDA do have to have some regulation, for example, validated gene, validated mutations, and each mutation from patient can be very different, right?
Yeah, exactly. That's why how does the regulatory approach has to be discussed continuously, but Peter Marks expresses optimism that if this effect, because it's a newborn baby and it's a lethal, almost life-threatening, it's not lethal, but it's 50% of baby will have life-threatened in this urea cycle disease. So that's why it's a huge motivation for the industry and FDA to work together, because this is not sustainable. For this paper, you know that how many, just one IDT have 12 co-author on the paper, because they have to work around the clock. That's just, it's not sustainable in terms of cost.
Okay, so that's great. Another question about the repeated infusion of the drug product is perhaps related to the percentage of liver cells that can be engineered, that can be converted It's not 100% rate, right? And with each dose, you saw increase the rate of conversion, right?
Yeah, that's a very good question because you're lucky here. LNP is mainly enriched in the liver, especially in the portal hepatocyte. So they do have even better than when they do the animal testing, because once you put a human, the percentage increase. But as you said, it's not 100%. That's why they still need to have some additional detail to work out. But they're lucky it's liver. For anything beyond the liver, can you do this? And Peter Marks is saying you need more research for non-liver tissue.
So fetal liver can be more easier for LNP. You mentioned that is that.
Yeah, because the LNP not only going to the liver highly enriched, it's going after some hepatocytes mainly. So that's really a good news because that's the activity of the urea cycle usually. So it's a really tailor-made. This disease is really tailor-made for the first proof of concept. That's why people getting so excited. But for other disease, maybe some disease is easier, like the fetal ketonuria, any Mendelian disease involving one nucleotide change, usually probably will work out quite well. But you also need to be addressing the manufacturing issue, the clinical batch, the batch consistency. There's a lot of questions, but definitely this is a landmark in the history of CRISPR, more exciting than Casgevy, I have to say, because Casgevy is ex vivo CRISPR. This is in vivo. This is in vivo base editing. There's so many unknown to deal with, but this is the first that is showing very promising sign for in vivo base editing.
Yeah, it's very exciting. Any comments? Any further comments on these topics? So maybe we can move forward because right now it's 10:30.
Yeah.